Alright, so we'll be going over the different cell organelles today. Pretty straightforward stuff, mostly high school level, but good to have as an introduction topic. So firstly, there's the nucleus. Everyone knows what this is. It's basically the command center of the cell. So that's because it contains cellular DNA. Another function that it has is that it controls protein synthesis. That's directly linked to the fact that it contains cellular DNA for the creation of mRNA and so on and so forth. Now it has some components within it which we should speak about. Firstly, it's the nuclear envelope. Nuclear envelope. So this is basically a double membrane which controls any exit and entry into the nucleus. Then we have something which we call the nuclear pore. And the nuclear pore regulates ion concentrations, and it allows for the movement of ions and things like that into the nuclear area. Next, we have something which we call the nucleolus. Now, this is like a small region within the nucleus, and it's basically the site where rRNA is formed, and that's used for the creation of ribosomes. And then lastly, one more thing to know is the chromatin. That's basically the arrangement of the DNA in our cells. So that's a pretty basic, straightforward review of the nucleus. That is the TEM version of it. You can see all the locations very clearly. As you can see, the nucleolus is a really, really tiny space. It's a bit darker, and that's about it. And that's the chromatins over there. Let's now move on to the mitochondria. One other thing to actually note before we move on to mitochondria is that the chromatin actually has only about 30% DNA. So you may be wondering what the other 70% is made up of. So DNA is just like the genetic material that we have, right? The other 70%, about 10%, is made up of RNA, and the other 60% is made up of histones, which are globular proteins which associate themselves with DNA, and they take part in their main function is to allow for modifications and things like that, and gene control basically and expression. On to the mitochondria. So for the mitochondria, you just add an image here. So that's a GM image of the mitochondria. I'm just going to put it on the side. So as you can see, this is the mitochondria. Now it has three areas within it: the cristae and the matrix, and then the outer membrane, basically outer and inner membrane, so the mitochondrial membrane as a whole. So firstly, let's just talk about its functions and things, and then we'll go into the specific suborganelles. Okay, so the first function, which is like the pretty obvious thing, everyone knows this, is like the powerhouse of the cell, right? Everyone says this, powerhouse of the cell. But we need to be a bit more specific as to why it is this. That's because it is involved in aerobic respiration, which is also known as oxidative metabolism. It uses oxygen. All right, and also obviously as a result of this, it is involved in ATP production, and about 95% of energy is provided by this. Pretty significant. Obviously helps in like day-by-day activities, one of the most crucial things. Now, another thing to actually note about the mitochondria is that it has its own DNA, which is pretty rare. Only the mitochondria and one organelle in the plant has its own DNA. But it's a bit, so there's two forms of DNA. There's 80S and 70S. And this basically just refers to the density of each of them. 80S is more dense and is usually present in the nucleus, things like that. And it's the eukaryotic variant. 70S is less dense, and it's present in mostly prokaryotes, but also in the chloroplasts and the mitochondria. So in the mitochondria, you have these 70S DNA things, right? Yeah, so that's one other thing to note about the mitochondria. Now, in terms of the organelles, like suborganelles, there are three. One's the matrix, Now, if you just have a look at what that is, it's like the inner liquid area. So the way we describe it is liquid-filled matrix, and this contains enzymes and things for respiration. So really important, obviously. And then the cristae, which is the other thing. It's these things on top. This is a cristae, this is a cristae. So these are involved in increasing the surface area. Increase the surface area. And then finally we have the membrane, the double membrane. And actually one thing to note about this, other than the fact that it's involved in entry and exit, which we just mentioned, involved in controlling entry/exit. One other thing to actually note is that the cristae, which we just spoke about, they're extensions of the double membrane. Cristae are extensions of membrane, of inner membrane, actually, not the outer membrane, of the inner membrane. So that's the mitochondria, pretty straightforward. I'm going to make this a little bigger so that it's in proportion. It's better. So that's the mitochondria, that's the nucleus. Let's now move on to the next organelle, which is the endoplasmic reticulum. There's two kinds of that. There's the soft and the rough. I'm just going to pull it up right now. Alright, so next are the endoplasmic reticulum. There's two variants. There's the soft and the rough. Rough is obviously these ones over here, and then the soft ones over there. The smooth, sorry, not soft. So let's just get into a little bit about their functions, and we'll also talk about the ribosomes in this section because they're actually pretty closely linked. But firstly, let's talk about the rough endoplasmic reticulum, but let's just title this as endoplasmic reticulum. So there's two variants, as we know. The first one is the rough endoplasmic reticulum. Rough. I'm just going to write ER. That's endoplasmic reticulum. Now this is involved in protein synthesis, and that's a huge process we'll get into later in biochem. But just note that it's involved in that process. And in addition to that, it contains ribosomes. Because ribosomes are the organelles involved in protein synthesis. Their main function is protein synthesis. Now, we'll get into what ribosomes, like a little bit more about ribosomes themselves now, but those are the two things to know. The way I remember that the ribosomes are present in the RER is that RR, so ribosome, rough endoplasmic reticulum. My way of remembering it, hope that helps you. Let's talk a bit more about ribosomes. So ribosomes are, as we spoke about before over here, we said that the nucleolus over here is where rRNA is formed. Now, rRNA is actually used in the formation of ribosomes. So two subunits, that's the other component of the ribosomes, plus RNA. So that's ribosomes. And then lastly, we have this smooth endoplasmic reticulum, which is a little more extensive in its functions. So we'll just write all of them down. But one of the main things it does is that it synthesizes lipids. Lipid synthesis. Now, if we go into a little bit more detail about the SER, is that, well, firstly, we just said lipid synthesis, right? It's also involved in lipid metabolism. And one thing to actually note, which is quite important, is that it's involved in the formation of lipoproteins, which are components of lipids, and basically in the liver. So you find a lot of this in the liver. Then, secondly, we have another really important function is the synthesis of steroid-based sex hormones. Steroid-based hormones as a whole, but an example is sex hormones, such as sex hormones. So examples of this are your testosterone, your estrogen, so on and so forth. Next, we have our another function, which is it's mainly involved in lipids, as you can tell. Another common theme: absorption, synthesis, and transport of fats. And this happens in the intestine, so intestinal cells. Another function of it, now this is involved in the liver again, along with some other organelles, which we're going to get into very soon, involved in the breakdown of and detoxification of drugs, such as alcohol, detoxification of drugs, mainly alcohol in the liver, and even carcinogens, actually. And then lastly, again, this is again in the liver. As you can see, pretty common trend is that it's mostly in the liver because it's often involved with detoxifications and things like that, but also this, which is breaking down the stored glycogen to form glucose to increase blood sugar levels when they are pretty low to form glucose. So those are the functions of the smooth endoplasmic reticulum, and as you can see, these are the DEM visuals of the SER, and then these are the DEM visuals of the RER. So that's about it for the endoplasmic reticulums. We'll get back into them in detail later on when we go to specific organs, right? But that's like an overview. Let's now move on to the Golgi apparatus, and then there's two others after that, the lysosomes, peroxisomes, and then we're pretty much done. Alright, so the Golgi body, as you can see, this is what it looks like. It's a pretty we've all seen this before, and it's basically made up of these pouches, if you will, and they're called cisternae, and they're basically pinched off from the reticulums, the endoplasmic reticulums, and that's how they're formed. And in terms of their function, well, the easiest way to think of it is that they're like the traffic directors of cell, and that is to say that they are involved in the sorting, packaging, and modification of proteins and lipids made in the ERs, so the endoplasmic reticulums. Now specifically in terms of modification, we can say that it adds carbohydrates to proteins to form glycoproteins, and it also does that with lipids. Add carbs to lipids and proteins forming glycoprotein lipid. Amongst a lot of other modifications, but that's the most like straightforward one that I can think of at the moment. So that's basically the function of the Golgi body. Of course, there's a lot more to it in terms of like the specific modifications and how it does the sorting and packaging. You don't need to know that at the moment, but this is like the basics of it. Let's now move on to the lysosomes. Now for this, the image isn't really super clear, but I'm still gonna add it anyways, just so you have an idea. Let me just write it down first. Lysosome and the image. So the lysosomes are basically what we call like the dustbin cell. Now that's not like an insult or anything. It's just that they break down and they break down a lot of cells and non-useful tissues and things. They're very, very useful, you know, and as such, they're mainly involved in the breakdown of different cells, basically, and tissues, things like that. But the main thing to note is that they're formed at the Golgi apparatus. And I guess a better way to think of them is like they're the demolition crew. They just break everything down. Very helpful though. But in terms of how they do it, they have an intracellular digestive enzyme. Acidic pH, and that's called lysozymes, and they're present in like a lot of your excretions. Like for example, in your tears, lysozymes are present in your tears and your sweat sometimes as well. Things like that. Now, the specific functions of lysozymes, there's a lot. Lysosomes, there's a lot, but their main thing is that they break down and digest dead organelles. Digest, break down dead organelles. But then they're also involved in breaking down like non-functional. So I'm just gonna keep adding here non-functional organelles. Even like after phagocytosis, the digestion of the remaining particles left by the phagocyte is usually digested by the lysosome. So digest phagocytic residues, I guess, or I don't know, that's one way of I can think of mentioning it. Even worn out cells, worn out organelles and cells, sorry, worn out cells only. They're also involved in break down bone, and that's used in that's done to release calcium in the blood, which is used in muscle contraction, amongst many others, but that's like the main few functions I can think of. Basically, they break down old, worn out, dead, non-functional, or harmful particles in your blood in some way or the other. Now, it's sometimes secondary, sometimes primary, and sometimes they do it themselves. Sometimes they come in after someone else does the work. Like for example, in the case of phagocytosis, where it's firstly engulfed by the macrophage or neutrophil, and afterwards they come in. But sometimes it's direct, like when there's a worn out cell, when there's, you know, they just like keep it clean, keep it tidy, you know, ensure everything's working optimally. And now lastly, one other organelle, and this is similar to lysosomes, and actually they're called peroxisomes. Now these guys are actually really similar to them in some senses, that in that they are also involved in the breakdown of certain things, but more specifically they are more useful in detoxification in the liver of alcohol and things like that. Now let's talk a little more about what they are. They often look like smaller lysozymes, but they're self-replicating organelles, and they do this by like pinching off of each other. Basically they pinch into half each time, basically pinching off each other. But like how we spoke about the lysosomes and how they're formed by the Golgi apparatus, by the budding off of the vesicles of the Golgi apparatus. It's not the same. It's just that they pinch in half and half and half over and over, and they're self-replicating, so they can form more of themselves in that way. And peroxisomes, basically the way that they work is that they contain really powerful enzymes. Now you may be wondering what these enzymes are. The two which are to be noted for detoxification are oxidase and catalase. And of course we'll go into more details on specifics in biochem later. But these are involved in detoxifying substances such as alcohol. And they also do this thing in which they break down and neutralize ROS, which are reactive oxygen species. So that includes your hydrogen peroxide and things like that. So hydrogen peroxide, which are very harmful for the blood as a whole. And they're obviously present a lot in the liver because they're involved in detoxification, present in liver and even in the kidney. So that's an overview of the major membranous organelles. Now there's also the cytoplasm and the cytoskeleton and all that, and we're going to get into that in the next one, because that's actually a pretty long one, and there's quite a few aspects to cover, from the cytoplasm to the cytoskeleton to different tubules, and then there's also the different filaments, and then there's the cilia, flagella, all that. There's a lot to cover there basically, so we'll get into that in the next one, but that's about it for this one. Hope that